Hello everyone, I am Dr. Sonali and today we are going to talk about a very interesting, a very important, a small topic that is Erythroblastosis Fetalis and Coombs test. Now let's see the battle inside the womb. Before we start, let's quickly revise the very, basic and the common concept of your blood grouping. Now we have the four common blood group A, B, AB and O. Now this can be A positive, B positive, AB positive, O positive, or it can be A negative, B negative, AB negative and O negative, right? Now this positive and negative is determined by something known as RH factor. Okay, that means if it's positive, there is presence of this RH factor, which is also known as the D antigen, okay, on your RBC. If it's negative, that means there is absence of the same, right? So your RH factor or your rhesus factor, it is a type of protein on the outside of your red blood cells, okay? This is the red blood cells, this is the RH factor, it is RH positive RBC. This one doesn't have the RH factor, so this is RH negative RBC. Okay, now let's see this pregnancy. This is a very calm, very uneventful pregnancy till now. Now in this, the mother is RH negative, any blood group but RH negative. Her husband is RH positive. Okay, again any blood group, but we have to focus on this RH positive and negative. The husband is RH positive. Now if the child was RH negative, like the mother, we wouldn't be having this discussion in class. But this child is RH positive. That means this fetus blood will have RH positive RBC. That is RH factor. This RH negative, this mother's blood will have absence of this RH factor. Okay, now let's see what happens till delivery. Till delivery, everything is fine. Okay, till delivery, this is a very uneventful pregnancy. This is the first pregnancy. Okay, so this delivery, the flow of blood is from mother to fetus. Okay, this mother having RBC without the RH antigen, that is RH negative RBC, going into the fetus, and it's just unidirectional flow of blood, everything is fine. But something happens during delivery. During delivery, there is mixing of blood. Okay, that means the fetus's blood get mixed with the mother's. Okay, the fetus and mother's blood get mixed. So what will happen because of this? So because of this, the mother doesn't only receive the baby, she also receives the baby's RBC which have RH factor on it. Okay, so baby's RBC entering mother's circulation, and these RBCs have this RH factor. Okay, now for the mother's immune system, this is something foreign. Now anything which is foreign, what will happen? There will be production of antibodies in the mother's blood. Okay, so this is anti-RH antibody, and this is IgG type. Clear till now? Now let's see what happens during the second pregnancy. Okay, now this first pregnancy, the baby got out just in time. The baby is fine. Now this mother got pregnant the second time. Again, in the second pregnancy, if the baby is RH negative, we would have ended the class here. But this baby again turned out to be RH positive. Okay. Now, what will happen? These sneaky antibodies which are present in the mother's circulation, these antibodies will cross the placenta. This anti-RH IgG antibodies crosses the placenta and binds to the fetal RBC having the RH factor or RH antigen. Understood? Okay. So what will be the result of this binding? The binding leads to breaking down of the fetal RBC, that is hemolysis. Okay. Now, what will happen because of the result of all this? There can be in utero death. Okay. Or if the baby is born, the baby will be suffering from severe jaundice and severe anemia. Okay. Both is the result of hemolysis due to breakdown of RBC. There is severe anemia, and this broken down RBC releases bilirubin, right? So this bilirubin will cause severe jaundice. Clear till now? Now this whole thing is called erythroblastosis fetalis or hemolytic disease of the newborn. Okay, it's a very common short note in your exams. Now, do you think this could have been prevented just after the first delivery? Well, it could have been. So if the mother is RH negative, her first, this mother, okay, RH negative, her first pregnancy, if the first pregnancy, the baby is RH positive, just after the delivery, what do is we give this anti-RH immunoglobulin injection, okay, immunoglobulin injection to the mother, okay, to mother. Why is this given? So that this anti-RH immunoglobulin clears up the fetal RBC with its RH factor before the mother's immune system notices and produces antibody. Understood? It is rapidly cleared the fetal RBC which she received from the first pregnancy. So no antibodies are formed, and the second pregnancy, if the fetus is RH positive, is fine. Clear? Now, this is all happening inside the mother's womb, okay? But we are sitting in the lab. Now, if I have to think about it, how to diagnose, sometimes apart from being a doctor, we also have to think like a detective. So let's take out our magnifying glasses and see how to detect this. We can detect it by Coombs test, okay? Coombs test is of two types. First is direct Coombs test. Now see what's happening in direct Coombs test is the erythroblastosis fetalis directly affects the fetus, your baby. Now if we are directly taking the sample of the fetus, it's a direct Coombs test, okay? So you take the fetus's blood as a sample here, okay? So your fetus's blood is the sample which has here red blood cells with the RH antigen, okay? Now they are coated with the maternal IgG antibodies which has crossed the placenta, okay? Clear till now? Now we have to add something to this. We will add Coombs reagent. Coombs reagent contain anti-human globulin, which are antibody to this maternal IgG antibodies, okay? Antibody to an antibody. This is Coombs reagent. Now, if agglutination occurs, your direct Coombs test is positive. It confirms there is hemolysis happening, okay? Now let's understand this with a diagram. First, let's understand a negative Coombs test, okay? So I have taken this fetus sample, blood sample. Now, if the fetus RBC, it is not coated with the maternal IgG antibodies, and if we are adding the Coombs reagent here, okay, Coombs reagent which I am adding here. Now, Coombs reagent is an antibody which binds to the maternal IgG antibodies which coats the RBC. But here the RBC is not coated. As a result, this test will be negative. Okay. But what happens when the fetus's blood is coated with the maternal IgG antibodies. After combining it with Coombs reagent, the anti-human globulin binds to the FC portion of the maternal IgG antibody and giving a positive heme agglutination. This was direct Coombs test. Clear? Let's move to indirect Coombs test. Now, since direct was from the fetus, indirect would be mother's blood. Okay? There are only two parties involved here, fetus and the mother. If direct was from the fetus, indirect would be mother. Mother's blood is the sample. Now you have taken mother's blood as the sample. Now, what is in mother's blood? Mother's blood will have her own RBC, but we are not interested in that. This is of no use to us, okay? So we will see there is something else also in this mother's blood. She has this antibody to IgG antibody to the Rh factor. Okay. Now this is free floating. Okay, this is the antibody which is present. We need this antibody to bind on something, right? Then only we can see agglutination. That's why here we will add a commercial RBC, Rh positive RBC, because these are anti-Rh Ig antibody, right? So we will add RBC which are Rh positive. Now, if a serum contains anti-RBC antibodies, they will bind to the RBC. And then when we add the Coombs reagent, agglutination, this indirect Coombs test will be positive. That is, mom has the antibodies against the fetal RBC. This. Your first is the patient sample containing this antibody. Okay. Now, we are adding this commercially available RBC, Rh positive, with this D antigen, that is the Rh factor. Okay. Now, these antibodies they bind to this RBC, and after that, when we add this Coombs reagent, what happens here? This forms a cluster, and it shows as visible agglutination. Okay, there is cluster formation. Clear till now? Now, before we move ahead, let's understand one more point here. See, your anti-human immunoglobulin, which is present here, this anti-Rh IgG, which is there. Okay. Now, this anti-Rh IgG is in the mother's blood. It binds to the fetal RBC. This is what we have understood till now, containing the Rh factor. Now, you see, this for visible agglutination to happens, there need to be clumping, okay? But this IgG antibodies, this maternal IgG antibodies bind to only one RBC at one time, okay? But for agglutination to happen, we need antibodies which bind to more than one RBC at one point. That's why we add this Coombs reagent, okay? This anti-human IgG is added, which you see here is doing what? Binding to more than one RBC at given point. It is forming a cluster, okay? This cluster is a visible agglutination. Since this anti-Rh IgG cannot do this, we also call this blocking antibodies or incomplete antibodies. Okay, clear? Now let's quickly see the difference between your direct Coombs test and indirect Coombs test. Sample is baby's blood. Indirect, the sample is mother's blood. For direct Coombs test, we detect antibody already on RBC. Indirect Coombs test, we detect free antibodies in the serum. Direct Coombs test it is used for diagnosis of your hemolytic disease of the newborn, but indirect is as a screening in Rh negative mothers in their second pregnancy. Okay, timing is after birth. Indirect is done during the pregnancy. Okay, and this will be done in their second pregnancy if the first pregnancy was Rh positive. Clear? Okay, this was all. Now, this is a very important topic almost in, every year, be it in your physiology, be it in microbiology, pathology, gynecology. This erythroblastosis fetalis is a very regular question. Even for your MCQs, you will get a lot of MCQs, okay? I hope this was helpful. Thank you.